Disclaimer. The following hour of programming may not represent your views or opinions. But what are your views anyway? Where did you get them? Did you personally select your views, or have they been selected for you by others? And are your preconceived secondhand views a good enough basis to form an opinion on what you're about to hear? The short answer is yes, you are perfectly entitled to form opinions based on other people's views. The long answer is written in several languages and overwhelms Google Translate's ability, thus leading to many flawed and potentially fatal worldviews, none of which should be recommended while operating a mixed drink. But to paraphrase it, it usually goes something like this. If you don't think for yourself, you are not being yourself, and therefore do not exist. And the following hour of programming is each week dedicated to touting the accomplishments of people who excel at long answers as well as thinking for themselves. The scientists. For no matter what views or opinions existed in their minds before the day's work begins, the mission of the scientist is to ignore it, at least long enough to produce something brilliant for This Week in Science. Coming up next.
I've got the kind of mind that can't get enough. I want to learn everything. I want to fill it all up with new discoveries that happen every day of the week. There's only one place to go to find the knowledge I seek. I want to know what's happening. What's happening? What's happening this week in science? What's happening? What's happening? What's happening this week in science?
Good science to you, Kirsten.
Good science to you too, Justin. Happy Thursday.
Thursday. Howdy, Blair.
Hello.
Hello, Blair. Blair's
Happy
back,
to be back.
from your international travels, and you brought back an international bug.
Yeah, I suppose I did.
You've gone pandemic.
Don't put me in a plastic room, please.
Yeah, we have. I told Blair to stay home tonight so that we wouldn't have contagion on our hands. Just Skype in, honey. Welcome, everybody. This is This Week in Science. I hope everyone's ready for a great hour of science news. We've got all sorts of stories ahead in this show. A whole hour of science news. Nothing but the stories for you and us talking about it. To start out, though, I just want to say, just to get started, it is on the day that we are recording this and broadcasting, it is International Women's Day. So I just want to give a very quick shout out to all the science-y women out there and all the women in my life, family, friends, teachers, mentors, everyone who's ever had a role in me becoming who I am. I just want to say thanks. And, for all the women who are working in the sciences, thank you for pursuing science because it's always worthy. Just want to put that out there. Justin, what stories did you bring this week?
Oh my goodness, what have I got? I have got Essence of an Ape. They've done some decoding on another ape species, bringing the total, I think now to 4, maybe including us 5. New way to ruin a relationship with that certain someone. Brand new. Science is always finding new and better ways to break up with your significant ones. This one will be pretty fun. And depending on the flow of the show, we could go a couple different directions. Faster, more accurate clocks. Researchers who've AIDS may have been cured this week. There's a couple stories out there. We'll see where we end up.
Wow, there's some big ones. Absolutely big ones. Blair, what did you bring today?
I brought a story about snow leopard poop. Yeah.
I'm looking forward to your animal corner this week. I was thinking about this story and I was thinking, well, if you can't catch an animal, just look at their poo, right?
That's what you got. Yeah, that's all you can find.
And I personally do have experience with snow leopard poop, so...
This is a very personally relevant story. Well, I brought some story about antimatter. Dark matter is just confusing the scientists out there, and some bad birds. And then there's some other stories. I'll give you a choice. Actually, I'm going to give everyone in the chat room right now a choice for the story that I'm going to get to last. It'll either be drug-free organ transplants, or spider strings? Your call. I'll watch the chat room and see what people want, and we'll read that one when we get to the end of the hour. But to start everything out, why don't we start with the things that make the physicists go, Things that make them go, dark matter is really confusing the heck out of physicists at the moment. San Francisco State researcher Andishe Madhavi and his colleagues have been looking at dark matter, specifically at the center of a galaxy cluster called Abell, Abell 520. And so 5 years ago, they imaged Abell 520 with a ground-based telescope. And what it told them was that there was, in this cluster, what they're seeing are galaxies moving through one another. And normally, the galaxies themselves, the mass that we see, it's so far apart, it doesn't really have much of a collision. It doesn't hit much, but there's a lot of mass that is unseen, and that's the dark matter. And that tends to have a really dramatic effect. And so they're able to see the splitting apart of the visible matter from the dark matter. And so we've seen this in other galaxy clusters and these intergalactic collisions taking place. But with Abell 520, things didn't quite make sense. So looking at the way that the visible matter was situated versus the dark matter was all clumped in the middle of these galaxies. And they 5 years ago said, wow, this just looks funny. And none of the simulations that we have predict this. Nobody says that we should find this anywhere in the universe based on all the ways we've looked at galaxies running into each other, on dark matter interacting with stuff. We just shouldn't see it. And so they're like, okay, we need time on Hubble. We need a space-based telescope to look at this to make sure that what we're seeing is really what we're seeing and we're not getting some distortion from the ground-based telescopes. So that's what they did. They went to Hubble, and they took a look. And so
to the Hubble-mobile.
To the Hubble-mobile, exactly. Intrepid space scientist detectives. And so there's this wonderful statement from an article from San Francisco State where Madhavi is a researcher. The 2000 study was, quote, a result that basically everyone wished would go away. And so this new study, it's published in Astrophysical Journal, shows, quote, without a doubt that there is a dark matter concentration in that piece of sky. So confirmed that there's dark matter clustered, but it doesn't explain why it's there. Their study has not figured anything out. It's just confirmed the 2007 results. So instead of that result going away, it's even firmer. So Madhavi is suggesting that our understanding of how galaxies grow and how they collide is incomplete, and that which isn't a far stretch. We've only gotten our understanding of how galaxies do interact from computer models, from simulations in computers, and so maybe they're missing something. Maybe we need more evidence that we can plug into these scenarios. Alternative, though, is that a new theory of dark matter interaction is needed. Not necessarily the most parsimonious of the two alternatives, but
parsimonious.
Parsimonious.
Can't just throw the big words at me, Kirsten. You gotta highlight the big words.
Not the simplest. Parsimony, the parsimony, principle of parsimony. You want to. The simplest answer is often correct.
Oh.
So the researchers are really trying to figure things out. And so Madhavi goes on to say, the only way we understand how galaxies grow up is with supercomputer simulations. My colleagues tell me the likelihood of spotting an oddball like Abel 520 is nil, but now we have the responsibility to go and do the hard work to check the simulations. And so maybe, who knows? Who knows? The researcher himself, he says, I'm just as perplexed as I was back in 2007. It's a pretty disturbing observation to have out there.
What an attitude.
He's
It's a
disturbed.
disturbing observation, and now we're going to have to go back and check our simulations. Oh,
Something's wrong here. Yeah.
Just, you don't understand. It's not that. I love that there's something new in space. It's a fantastic discovery, but you don't understand working with these computers. It's like it's guaranteed like another 250 hours at a keyboard making this simulation work. And that's just going to be annoying. This is now my life.
Yeah, it's kind of funny. But anyway, I love the mystery around all this where, you have an observation that doesn't quite fit the models. And so the researchers are like, all right, so our models might be wrong. We need
just
maybe
fantastic, right?
we have to
Because they should
look
be
things
wrong.
more deeply.
If we have everything that figured out, where we can do a computer simulation based on just not having a lot of observation on this, that's the toughest time to make a simulation of anything. You're simulating something you haven't really seen take place, so you've got a lot of factoring. It probably wasn't completely off. I don't know, but now you have something observable, and this is why it's always important to have experimentation, to have these telescopes, to have all these things in place, because not finding what you expect to find is the greatest result, because then you can take that information and plug it back you can work backwards from that, figure out why you got to where you did on your simulation, and work backwards from your observation. And at some point you'll meet in the middle. And there's always the most fascinating thing when this was very much Einstein's approach to things. He'd have an idea and he'd start going on that direction on paper, and then he'd find there'd be an observation of something in the world, and he'd try to work backwards to see how he could connect the physics that he was familiar with, or that he was creating for him in his case, really, and the observations that he was making. And then when you finally get to that point where you can connect the two, then you have a really powerful working model and can begin to play with it and dissect it and figure out why it's working the way it is.
Yeah, well.
So yeah, this is a huge breakthrough for dark matter.
Yeah, it's huge. It's wonderful. So there will be more to come. The scientists will not sleep until they have solved this mystery. In other crazy physics news, we've talked a lot about antimatter on the show in previous episodes, and researchers at CERN in Switzerland have published a paper in Nature. It's from the Alpha Collaboration, which are the group that are trapping antimatter. One of the researchers is at UC Berkeley, and where's another? We have Joel Fajans at UC Berkeley, and then where else are the researchers located? I'm forgetting. Alpha, Wortel, Berkeley. I don't know. Berkeley. I'll just keep saying Berkeley, even though I know there were more groups involved than that. Anyhow, so first they trapped antimatter using super magnets. They were able to trap antimatter in this, what they call a bottle. Excuse me. That was a sneeze. Thank you. So they trapped, maybe I've got the plague. Blair, it's better that you didn't come over here. So they trapped antimatter first off. Then they trapped it for like 15 minutes, and they trapped multiple antimatter atoms. And so now they know that they can do it, and now they're going to start looking at the antimatter, testing it, trying to see what its properties are. So they've been trapping antihydrogen, the corresponding particle to matter's hydrogen. We all know the wonderfully simple, it's the basis for so much of our understanding of atomic physics, the hydrogen atom. Well, they're looking at the antihydrogen atom to determine exactly how it compares. Does it act the same as hydrogen? And so they have published a paper in Nature that describes the methods that they're using to determine whether or not they can actually observe properties of the antihydrogen. And in fact, indeed, they can. And they're really excited about it. So next step is I'm sure we're going to be seeing a paper from them in the near future. Give it a couple of months. We're going to be seeing whether or not antihydrogen glows in the same color as hydrogen. I mean, that's the first step. What spectra does it emit light at when it is induced to do so? Does antihydrogen look like hydrogen? Yeah, they're very excited about it. If
you fill a balloon with it, does it go down into the earth?
Right. So Fajan says we have every reason to believe that hydrogen and antihydrogen are identical, aside from one being made of matter and the other of antimatter. In fact, CPT, which is charge parity theory, insists that they are identical. Oh, sorry, not T. The T is not theory, time reversal. Sorry, charge parity, time reversal. So, in fact, CPT insists they are identical. But what if they are not? What? What if they're not identical? What? And that's going to change a whole bunch of more ideas. So, it's important. It's important that they look for differences, even if those differences are not there. It'll tell us a lot about the universe and why it is made of matter. Why are we here, made of matter and not antimatter?
You know what I'm afraid they're going to find someday? That it turns out that all this time, we are the antimatter. And we've been destroying the real universe.
Yeah, it's all in a name, right? I am my father. No, what?
Huh?
I don't know. Yeah.
That would be a real trick.
All right. What did you bring? Tell me some stories.
The gorilla genome has been decombobulated, and there's a multinational group of researchers. This is the last genus of living great apes to be decoded. While it confirmed that our closest relative is still the chimpanzee, they haven't been booted from the spot. It did show something pretty interesting. It shows that there are sections, I think it's about 15% of the genome that is much closer to humans than we are to the chimpanzee or the gorilla is to the chimpanzee. So it kind of illustrates that we're not more evolved than our ape cousins. We've all evolved equally over the time that we've separated. There are sections of the gorilla's DNA that closely resemble ours. We have more of those in common with the chimpanzee, and a bunch that are also close to the orangutan. And then within those groups, we can see how they're different from each other too. The only thing that really separates us from the other apes or even other animals in general is we're the only animal that's completely insane.
Right.
That's really when it comes down to it. It's some sort of defect in humans. It just made us completely insane, self-aware, and that's how we ended up with all this.
Yeah.
This intelligence. It's not intelligence. It's some sort of. We should diagnose it someday with whatever we came down with. But it says here, the gorilla genome is important because it sheds light on the time when our ancestors diverged from our closest evolutionary cousins. It also lets us explore the similarities and differences between our genes and those of the gorilla, the largest of the living primates. That's Alwyn Scally, first author from the Wellcome Trust Sanger Institute. Using DNA from Camilla, a female western lowland gorilla, we assembled the gorilla genome sequence and compared it with the genomes of the other great apes. We also sampled DNA sequences from other gorillas in order to explore genetic differences between the gorilla species itself. So one more creature is now known to science. This is the preliminary. We'll probably be seeing a lot more of these in the future, and we may even get to the point where it can tell us which specific traits of ours are most similar to a gorilla? Which would be kind of interesting.
Yeah, I think, we are still distantly, we're related to gorillas. We're not super, super close, but it does make, it does, it's interesting to see that we're more related, that there are certain genes that are more similar to the gorilla
so it's something
than
along the line
previously
of
thought.
15% of the genes are much more similar than gorillas are to chimpanzees or than we are to chimpanzees. So that means either chimpanzees took a different route. Well, I guess, yeah, they would have had to probably taken a different route or been borrowing from a different, it depends on how the split happens when all that, sort of takes place. But it's sort of more of a triangulation back on to what that ancestor would have looked like genetically. Because what you can actually start to do is work that puzzle backwards and say, okay, chances are we didn't evolve the same as a gorilla did in that 15% of the genes that are very similar. So those were likely shared with the ancestor. And those things that we share in common with the chimpanzee are likely shared with an ancestor. And you can go with compare the orangutan and the chimp, or the chimpanzee, the orangutan and the gorilla, and you can sort of start to triangulate the ones that are very much in common that are showing up, even if some have diverged in this area or the other, can sort of create a picture for what the original ancestor DNA would have looked like. So it's a really fun project that's going on in assembling all of this. It's really going to show. We'll actually perhaps be better able to know this ancestor than we know a lot of the living creatures on the planet today, because we will have a pretty good look at its genetic code.
Yeah, and something that I think is pretty interesting is information about what it can tell us about how our brain and our traits developed. There's one aspect of this story deals with the genes responsible for hearing in the gorilla, and there's a great similarity between those genes in the gorilla and in humans. And so it was thought that the brain area and the genes related to hearing had to do with our development of language. And so that they developed in a particular way very rapidly because they were developing along with our rapid development of language. But to see that gorillas that do not have language the way that we do have the same or very similar genes that also radiated at a rapid pace. Shows that there's something else that went on in how that trait of ours came about. And so what we think currently is not right. And so it kind of sends ideas back to the drawing board so that people have to figure it out. Get back in there and figure more stuff out.
Oh, but that drawing board room is so cold. It's never worked. And then in the summer, it's hot. The air conditioner's all right. Still, that guy still kills me. It's really disturbing. How could that possibly be disturbing to you? You're a scientist. It's so exciting. Found something new. Maybe he meant disturbing in a good way. Maybe he said it really smirky, right? It was like, ooh, this is disturbing. Yeah. Right.
Oh, my goodness. All right, Blair, tell us a little bit about the animal world. It's time for Blair's Animal Corner.
Yay!
Blair's Animal Corner.
Oh, do I get a theme song? Ooh.
That's right. You're going to need one.
So I brought a story about snow leopard poop. Snow leopards, they live in the Himalayas, and they're really hard to research and study. They try to set camera traps, they try to observe them in the wild, and they're just a big mystery. And a lot of the conservation efforts with them are involved with the native peoples trying to kill snow leopards because they think they're taking their livestock and they're eating them. So that's what's really interesting about this fecal study, is that they did DNA coding in the feces. They looked at mitochondrial DNA in the feces, and they could see what the snow leopards were eating, which is great because we had no definite idea of what these guys were eating. The native peoples were pretty sure they were eating their sheep, but we weren't.
I think they're
that
eating my sheep.
clear.
Nobody's
Yeah.
seen it, but something ate my sheep. It's got to be the snow leopard
Or the
It's gotta
chupacabra.
be.
One of the two.
or the chupacabra.
So from the feces, they found out that 70% of their diet is Siberian ibex. And then domestic goats and sheep are the remainder pretty much of that. So the goat was 70%, the sheep was 8%. The rest of it, they weren't quite sure what it was. But essentially, that means
They are eating the sheep,
they
but
are,
not as
but
much.
not as much. They're eating a lot of wild ibex, and in fact, they were saying all of the wild ungulate species that they looked at were almost 80% of the feces of the diet that they were consuming. So they are eating some of these domestic sheep, but if they were going solely for these sheep and they were stuck in these pens and they were chowing down on them all the time, that would be most of their diet. If I'm a wild snow leopard, I'm not going to spend hours hunting an ibex if I can just walk up into a pen and eat a sheep. So it doesn't look like they're actually stealing that much livestock from these native peoples, which is really interesting to me.
I wonder if as they're figuring out the percentage of the diet, of course, they're taking all the samples of scat that they're finding around and lumping it all together into this statistical number. 70%, but what does that tell them about individuals? So are there certain snow leopards that prefer ibex? Are there certain snow leopards that are much more, I want a sheep Big Mac,
That's a very good point. And I think that is probably the next step of this, is that they should be looking at each individual snow leopard and looking at their percentage makeup. And when I've went to some events by the Snow Leopard Conservancy, a lot of what they do is they're trying to educate the native peoples on how to house their sheep so a snow leopard can't get them. A lot of them actually will only build 3 sides of a pen, and the 4th wall of the pen will be the wall of the mountain. And for a snow leopard, that's just like a ramp into the buffet of sheep, So
Yeah.
It's not the best tactic, even though it might be the cheapest and the easiest. So that's a lot of what they do, is they come up with ways to house the sheep where you can cover up the top of the pen so that the snow leopard can't get in. So that it would be interesting also to see if we adjust that, and the more that the native peoples are educated, if the percentage changes then too. That would be interesting to see, I think.
Yeah, I think it would be. How long, do they have any details about what it was like to go collecting the snow leopard poo?
I did not see much in the scientific paper about picking up the feces and what maybe the consistency was. I can tell you from my experience that snow leopard feces is pretty hard, and it looks like harder dog poop, I would say. It's not gushy. It's not that gross, really. I don't know.
Wow. See, they should take me out next time on one of these. If all they're finding is the poop to study, unless that's the only thing they're looking for. Because I've gone in search of snow leopards on nearly a half dozen occasions and have found them, have seen them every single time.
Wow.
In zoos?
At the Sacramento Zoo, yeah, they've got 2 snow leopards
impressive.
over there.
Yeah.
And I know right where it is. It's not a huge zoo, but it's kind of tricky the way it doubles back on some of the paths. So, yeah, if you don't know where to start, you could miss them.
You could totally miss them. But I do want to say, you mentioned the Snow Leopard Conservancy. They do some amazing work, and I think we've actually, several years ago, we spoke with somebody from the conservancy, and they're really working hard to try and maintain the populations of snow leopards that are out there. And so you should definitely, if you're interested, look for information on them, because they're a great organization. And Sacramento Zoo in Sacramento, it's once again a very great organization. Disclaimer: I know somebody who works there, but it's a good zoo for an inland.
You know somebody
place.
who works there?
I do.
Can they get me in to see the feeding of the big cats?
we could find out.
Because see,
But-
this is, and Blair, maybe you do this at the San Francisco.
Justin, wait, can we go to the triple shot here? Triple shot. Look
All right,
to
Blair.
look.
Yeah, I know. I did. Submitted to San Francisco.
San Francisco
Yeah,
Zoo, yeah.
Do you have feeding times where you allow the public to go in and watch the feeding?
We don't have set feeding times, but if you come in the afternoon, there's kind of a steady feeding and training in the afternoon. There's a pretty good chance you'll catch something if you come in when the lion house is open.
Oh, I get to catch my own dinner. Wow, that would be exciting. Well, maybe it was, because it's a childhood memory, but it could have been the San Francisco Zoo, actually. And I've always thought it was Sacramento, but maybe it wasn't because I've never seen them doing this there. But I remember there were times that were posted where you could kind of go in and watch the feeding, and they'd throw these big chunks of meat to into the lion cages, and the backside there was like a viewing area thing.
Yeah, for years and years, San Francisco had a 2:00 feeding, but we don't do that anymore.
Okay.
Yeah.
Okay, that's why I'm always like poking around like in Sacramento. I was like, I remember being back there and seeing them feed these cats, but I don't know how to get there. So that's why.
You know how we get there from here? We take a break. It's
We
time
go to
for
the
our
bridge.
break.
You gotta get on the bridge.
Get on the bridge.
You lined up a great punchline, then you blew it. Like, how do we get to San Francisco? We gotta take the bridge. That's how do we get to? Never mind. Okay.
All right, everybody, you're listening to This Week in Science. We are going to take a very short break, and after a few words, there'll be more science. So I hope you stay tuned.
Can't explain the things you heard with more than intuition. The line of reason shows the way to go. No conclusion. The methods of hypothesis and patience are the only things I need. Put on a pair of goggles and go looking for the things I couldn't see.
Twis would like to thank Audible.com for sponsoring this hour of This Week in Science. Audible.com is the leading provider of audiobooks with over 75,000 different titles in a variety of genres. And we here at Twis have enjoyed many a science-based book from their library and know that you can too. And to do so, Audible will help you out. Start a free trial. You get a free audiobook download if you sign up today. All you have to do is sign up at audiblepodcast.com/twist. That's audiblepodcast.com/twist. Go there now for your free audiobook download. And while we're talking about books, the Twis Book Club is reading books. Every month, we've got a new book that we read. And this month, our book of the month is Dirt, The Erosion of Civilizations by David R. Montgomery. So I hope that you would like to read along with the Twis Book Club and start a conversation about it. We have a Twis Book Club page on Google+. And additionally, you can go to our website and converse there. But have a conversation. Talk about the book. Dirt. It makes what's for dinner. It does. Dirt's everywhere. Twis also has merchandise that you might enjoy. So head over to our website, twis.org, to get yourself a copy of the This Week in Science 2010 Science compilation CD or a World Robot Domination t-shirt. We got some other t-shirt ideas in the works, but those aren't there quite yet. But if you want World Robot Domination shirt, a CD, that's right. Yeah, I said CD. I know I said it. Twis.org, it's where you go to get your hands on that merchandise. Additionally, Twis is supported by listeners like you. You make this show possible. You allow us to have the bandwidth and hosting that we need, contractors we need to hire, and sometimes to do some fun stuff. So it's your money that helps us do that. And we appreciate any amount that you're able to give. $2 to $2 million, $2 million, 2,200, 5. Whatever you can give. We make it easy for you by putting PayPal donation buttons on each episode page on our website. So if you go to twist.org and go to the most recent episode, you will be able to listen to the show if you want to make a few comments on the show and hit one of the donation buttons. You can do a one-time donation, or you can even do a recurring donation if that's more your style. But we ask you to do that. Twist.org. Donate. We thank you for your support. We really couldn't do it without you.
Our first assumptions were correct. Disprove the rest. It's hard. Methods and hypothesis and patience are the only things I need. Put on a pair of goggles and go looking for the things I couldn't see.
And we're back with more This Week in Science.
That's right. We are back, and I've got some science for you. How about some bad bird behavior? I want to stay on the animal tip for a little while here. Yeah, bad birds. Well, maybe they're just badly behaved birds. I don't know. But these birds of a species, yes, species Coracius garrulus, means they talk a lot, right? There's a paper in Royal Society Biology Letters that finds the babies of these birds puke on themselves when they're threatened. So they, yeah, these little babies, and this is
Well, you know,
a self.
would-
This is what were you going to say?
if I was looking to catch my dinner and it just vomited all over itself, I know I'm gonna have to clean it or no. Find
Exactly.
another one. I'm not gonna. That looks like a sick bird. Why would I eat that? I'm not gonna eat something that looks like
Exactly.
it's sick and dying. No.
And that is the whole point. So these birds, this is a self-defense mechanism. These little baby birds, they're pretty defenseless, other than the fact that they can throw up this fluid that is not just vomit, but it's foul smelling and orange colored. So it's this shocking color, and it smells really bad, really badly. And the researchers tested this cue by painting either lemon juice or some of the vomit on little baby birds in the nests, and then trying to find out how the parents reacted to it. Not necessarily that not necessarily seeing predator reactions, but seeing how the parents reacted. And so the parents, upon returning to the nests and smelling either lemon juice, which is not so bad, or the vomit, the birds that smelled the vomit took a little bit longer and acted more cautious in returning to their nest. So the parents, the hypothesis they were looking at was that this vomit also tells the parents that something happened while they were away, and to be cautious in returning to the nest. Maybe there's some predator like a snake or a rat or other animal still within the nest that they should be aware of. And so this not only defends the birds, but also helps the parents know that while they were out, there was some bad stuff going on.
I had no idea I dressed like a threatened bird. today.
Yeah, luckily we can't smell you.
yeah. You're lucky there.
Yeah. But there, this paper from the Australian Broadcasting Center, ABC, is that what it is? Center. It also talks about the northern fulmar, Fulmaris glacialis, that brings up stomach oils. stuff that's in their stomach. It vomits up these oils against intruders that makes them lose their waterproof coating. And then the common eider, Somateria mollissima, and the northern shoveler, Anas acuta, have the ability to spray feces on their eggs so that would-be egg thieves might not want to steal them. Yes.
Hey, whatever works in the wild, wild kingdom of being an animal, I guess, you go with it, right?
You go with it. That's right. So in this case,
I'm glad people don't do that, though.
Go with the vomit.
The nervous, like ink dye tag exploding orange vomit whenever somebody gets nervous, or the like putting feces on our young. It's just, I'm glad we figured out other ways.
Sometimes it's good to be a person, right?
Although I think it, it can work still in humans. I've been handed many a child with a full diaper and been like, oh, really? Like, I love my child, but is it my turn? Okay. All right. I'm on.
Yeah. What else you got?
Oh, this is, usually I try to avoid doing too many of the cancer potential cure, AIDS potential cure type stories, because there are so many of them. I mean, these are aggressively pursued diseases where they're looking for solutions. And so there's a lot of papers that come out finding another inkling, another thing, another direction that they could be going that sounds very much like a cure, but usually amounts to just needing to do more research. This is something, though, this one did catch my eye. So I'm going to go ahead and do it. This is a team of researchers at the University of North Carolina at Chapel Hill successfully flushed latent HIV infection out from hiding. They used a drug that is usually used to treat certain types of lymphoma. So one of the big problems with the HIV virus is that it has the ability to hide within cells and tissues of the body and then reemerge later. So most treatments of HIV virus, the antiretroviral therapies, are basically playing a game of whack-a-mole, right? They're really good at addressing the HIV virus when it pokes its head up and can smack it, right? But it's hiding throughout these tissues, and so reinfection can occur. And so really what you're doing with the antiretroviral therapies is reducing the load, right? You're not removing it completely, but you're reducing the levels at which the virus, through this pharmaceutical whack-a-mole, right? But what this has done, though, apparently, let me give you the quote here. Lifelong use of antiviral therapy is problematic for many reasons, not the least of them. Among them are drug-resistant side effects and cost. In fact, also, you got to use them for your entire life, right? So that's an ongoing expense. This is according to David Margolis, MD, professor of medicine, microbiology, and immunology, and epidemiology at the University of North Carolina, Chapel Hill. So what they basically did was they found, this is the first way to demonstrate the biological mechanism that keeps the HIV virus hidden and unreachable as something that can be targeted and interrupted in humans. So this actually eradicating the hidden virus within the tissues. The clinical trial, 6 HIV-infected men who were medically stable on anti-AIDS drugs received a vorinostat, an oncology drug. Recent studies by Margulies and others shown that vorinostat also attacks the enzymes that keep HIV hiding in certain CD4 plus T cells, specialized immune system cells that the virus itself uses to replicate. Within hours of receiving the vorinostat, all 6 patients had a significant increase in HIV RNA in these cells, evidence that the virus was being forced out of its hiding place. What that then, of course, allows is for all the heads of the whack-a-mole come up. Everybody knows what whack-a-mole is, right? It's that
whack-a-mole.
Right, it's that insanely old arcade game where the little rubber rodents pop out of the different holes and you have to hit them. They keep popping out in different spots. You got to whack them back down. It's as though all the rodents all go up at once and allow you to whack-a-mow them down, right? This proves for the first time that there are ways to specifically treat viral latency, the first step towards curing HIV infection, says Margulies. It shows that this class of drugs, HDAC inhibitors, can attack persistent virus, and it may not be the magic bullet, but this success shows us a new way to test drugs, target latency, and suggests that we can build a path that may. This is always the problem with every potential cancer or
may.
It suggests that we may lead to a cure, possibly, potentially. It's not there yet, but this is really good. This is a really good news one though, because this is of the HIV stories that we've covered over the years. This was the one problem in attacking HIV that has persisted against every form of treatment is the fact that it can hide. So yeah, get it smoking it out. Really looks like it could be a pretty big breakthrough in making the drugs that we already have out there much more effective.
Yeah, that's amazing. It could be absolutely amazing, and it could change the entire way that we approach the disease, which would groundbreaking.
Yeah.
Yeah. That's big. Did you say, can you say again who did this work?
University of North Carolina at Chapel Hill. Nice. And let's see, it was David Margulies, Margulies. Okay.
Cool.
Professor of medicine, microbiology, and immunology, and epidemiology at the University of North Carolina at Chapel Hill. You had it that time.
Nice. Very nice. Let's see. Worm fossils. Oh, worm fossils. So you were talking earlier in the show.
How can a worm leave I guess it's huh? Tiny worm skeletons being found? That would be remarkable.
Little worm skeletons. Well, in this case, it's not as shocking as it's not necessarily a skeleton, but that actually is kind of teasing the story a little bit. So these paleontologists have been looking at fossils that were found in the Canadian Rockies, and they found this old worm that used to swim in the oceans about 500 million years ago. And these little worms, Picala graciliens, is what they would call a primitive member of the chordate family. And the chordate family is so named the chordates because of the notochord, the vertebral column, the nerve bundle that runs the length of the body. And so the chordates include fish, amphibians, birds, reptiles, and mammals. This individual, most primitive known member of this family, would make it our oldest known ancestor. This is this little worm, this Picala species. This worm is the first individual that has been found to have a notochord that would become part of the backbone, and skeletal muscle tissue called myomeres. So they also found a vascular system as well. So it had many of the traits that would go on to become very important to vertebrates like us, vertebrates. Very important stuff. The most primitive chordate. The researchers say, Simon Conway Morris from Cambridge University says, the next time we put the family photograph on the mantelpiece, there in the background will be Picala. Interesting side note, first specimens of Picala were collected by early explorers in 1911, but they were overlooked as early ancestors of earthworms or eels because they didn't really seem to fit that category, just looking at them. And they're about 5 centimeters long, sideways, flattened, and it probably swam by curving its body side to side. Choop, choop, choop. Anyway, yeah. Little swimming worm. Your earliest ancestor, Justin.
I have many early. I'm sure I've got earlier ancestors than that. Come on.
Yeah, you do.
Come on now.
You do.
So this is
Like
actually this is perhaps the most dangerous story to come out in a while. Team at University of Buffalo State University in New York is working on video analysis software to track and decode eye movements in order to spot liars. You know, you thought those researchers working on a modified flu virus were perhaps tempting fate by creating this and making this public.
Yeah.
Imagine what will happen to our political system if all of a sudden we have a way of telling if somebody's telling the truth or not. The whole facade of a free and open democracy would just crumble immediately. So far, they say the results show that their software can spot liars with a promising level of accuracy. Their claim is based on their studying 40 people. Really not a huge case study. 40 people. Yeah. Their system correctly identified who was telling the truth and who was lying 82.5% of the time, which is more than enough to uncover many of the subtle white lies that we humans tell in the course of a day, course of a normal conversation, and thus could ruin even the most cherished relationships between friends and loved ones. What we wanted to understand,
especially if this technology is made into an iPhone app.
Is exactly what I'm thinking, right? Like you create like we could have it up on the Skype windows, like it'd be a Skype plugin. There'd be a little BS detector in the corner. Or yeah, you just download the app on your phone. It's like, okay, wait, tell me that again. I'm gonna see. I got my little pocket lie detector app on here. I'm gonna find out if you really think I look great in these jeans. Oh,
so what was the quote that you were gonna jump into from the researchers?
What we wanted to understand was whether there are signal changes emitted by people when they are lying, and can machines detect them? The answer was yes, and yes. Says Ifeoma Nwagu, co-author of the study and professor of the Center for Unified Biometrics and Sensors. It's like a real human approach. This is, according to Scientific American, their work was inspired by original findings from professor of psychology at the University of California, San Francisco, Paul Ekman, who has focused on emotions and how they connect to our facial expressions. As for the interrogators themselves, their experiences indicate that the use of such software for telling who is lying and who's telling the truth would not be practical in all instances and might not always lead to the right targets. Might not, because there's, it's got the false positives are in there too. And when you get into false positives, then it becomes really hard to actually use it in, say, a court of law or something like that. Although they are working with faster algorithms that may raise the software's ability to spot behavioral deviations in the real time or near real time, near enough to use, say, an app on your phone and interrogate your loved ones at home.
Yeah, the interesting thing, I think, is that the computers, though, in this study, even though they're only looking at 40 individuals, the 82% is better than experienced people at telling the lies. So people who are experienced in looking at these micro expressions in biometric signals of lying, that people are the experts aren't
the
as
mentalists.
Good at the yeah, exactly, the lie to me individuals. They're not as good as the computer program that's been created.
Right, and they are looking to expand this research and to continue with it. They're going to try to increase their sample size and see if their trend continues, because they even say that their sample size was too small to be statistically significant to really maybe not ready for prime time. But they're going to continue with it and see if it continues to work.
Yeah, and then you have to start thinking about the lying of people who are okay, they know they're lying. These individuals have been told to tell lies and just say a whole bunch of untrue things. But what about individuals who don't think what they're saying are really lies? So they are lying, but in their head, they don't really know they're lying. They don't think they're lying.
Yeah, well, the delusional would be tough. What I think would be more interesting is you'd have to create the
It's hard to catch a sociopath.
Well, right. Except that when people tell lies too, there's the real time lie where you're like, okay, now I'm going to say a bunch of stuff that isn't true, and my eyes are going to dart this way or that, or I'm going to have a different blink rate or something like that. They're looking at a whole bunch of different little matrices to come to these conclusions. But the other thing to look for is it's like a tell in poker. It's not the expressions that you make while you say something. It's sort of like the moments after you've gotten across that lie. There's a little relaxation that takes place, and like mission accomplished. I pulled it off, right? Yeah. Where there's like a little, for let me, I don't know that that the President Bush, right, former President Bush was a compulsive liar, right? It may have just been he was happy to get a line out without flubbing, but if you watch it, he does that a lot. Like he'll say something real earnestly, and then kind of give he gives himself a little half smirk smile going on there. Like, got that one out. And it doesn't mean he's lying, but it's the same sort of thing. It's like, I've accomplished it. I got that across. And there's a little relaxation in the mouth and the face and a little celebration thing that takes place after. So even when somebody is consciously telling you something that's not true and really trying to believe it themselves in that moment, there's still tells. It would be fascinating. I'm picturing this. Forget the iPhone app. Picture the next round, next election cycle. How they have all the graphics now on the cable networks. We're like election 2012. The graphs of people putting ballots and things and all these different like 3D-ish phantom computer graphics to simulate what's supposed to be election coverage. If they just had this computer analytical system software running in the corner of the screen, so as each person talked, it gave you a little BS detector meter. Like that would kind of like that would be awesome. But then, like I said, that would ruin our political system forever.
Yeah.
And do we want? Do we want honest politicians? Do we want honest people? Like people who can't lie? People who just do we want terrible liars? That's what the detectors work. We know you're lying. We understand you're making stuff up. You're changing your opinions on everything. But if you can fool the computer, you're going to be a great head of state because you'll be able to deal with the rest of the world in negotiating the messes that we make.
Well, we're getting to the end of the show, and I wanted to throw out a quick story. I don't know. It was pretty split earlier in the show where I asked whether or not people wanted to hear about spiders or whether they wanted to hear about drug-free organ transplants. And people said they'd had too much spiders. There were a few people who said there'd been too many spiders on Twis recently. And you know what? You can never have enough spiders. Spiders are awesome. So we're going with spiders, everybody. This isn't necessarily a spider story, but more a spider silk story. A Japanese researcher, Shigeyoshi Osaki of Japan's Nara Medical University, has been looking at the mechanical properties of spider silk. And so he took spider silk from female golden orb weaver spiders, Nephila maculata spiders, and he twisted between 3,000 to 5,000 individual strands of silk to create violin strings. And he then looked at the properties of these violin strings. And so there is an audio sample from on this story on the BBC website, just if you want to hear what a spider silk violin string sounds like. Alex can play that maybe really quickly.
1 second.
Okay, thank you very much.
Da-da, da-da.
I'm just waiting. I'll edit it out. It's easy to edit out this.
I'm looking for the
It's a black box that has a little volume thing and a little triangle.
Oh.
Hit the triangle.
Oh, the triangle.
The little arrow thingy. Yeah.
What is it on a cassette?
No,
Alex has never
The
seen a
page is loading
cassette.
very slowly.
Button, you have to press it all the way down. Don't hold the red button down with it.
Okay,
No,
wait,
you're
okay,
recording
it's-
over
Quiet.
it.
All right.
Rachmaninoff. Very nice. Very nice. I have no idea what that was. But if nobody else does too, people will say, wow, he called it as Rachmaninoff right away. Wow.
I bought it.
Yeah, right? Like, you can assume people are like, Bach. Yeah, Bach. That's one of my favorite concertos. But I have no idea. Oh, we lost Dr. Kiki. Well, do you have the, maybe we should just sign off. We should just end the show. Do you have the end of show notes? Shout outs at the bottom. If you get, it's at the bottom of our wiki. You can play the Kirsten.
Sure. Yeah, I got it.
Thank you, everyone, for listening. We hope you enjoyed the show. Twis is also available as a podcast. Just search for This Week in Science in iTunes, or if you have an Android device, you can look for Twis for Droid in the Android marketplace. That's Twis, the number 4 Droid, or you can look for Twis in the iPhone marketplace as well.
For more information on anything you've heard here today, show notes will be available on our website, www.twis.org. We also want to hear from you, so email us at kirsten@thisweekinscience.com or justin@thisweekinscience.com.
Be sure to put Twis, TWIS, somewhere in the subject line, or your emails will be spam filtered into oblivion. You can also contact us on Twitter at Dr. Kiki or at JacksonFly. We love your feedback. If there is a topic you would like us to cover or address, or a suggestion for an interview, please let us know.
And we will be back here next week and hope you'll join us once again for more great science news.
And if you've learned anything from today's show, remember,
Blair, this is all you.
It's all in your head.
This week in science. This week in science. This week in science. This week in science, it's the end of the world. So I'm setting up shop, got my banner unfurled. It says the scientist is in, I'm gonna sell my advice. Show them how to stop the robots with a simple device. I'll reverse global warming with a wave of my hand. And all it'll cost you is a couple of grand. 'Cause this week's science is coming your way. So everybody listen to what I say. I use the scientific method for all that it's worth. And I'll broadcast my opinion all over the earth. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 I've got one disclaimer and it shouldn't be news. that what I say may not represent your views, but I've done the calculations and I've got a plan. If you listen to the science, you may just yet understand that we're not trying to threaten your philosophy. We're just trying to save the world from jeopardy. Jeopardy. And this week in science is coming your way. So everybody listen to everything we say. And if you use our method instead of rolling a die, we may rid the world of toxoplasma gondii. Cause it's This Week in Science. This Week in Science. This Week in Science. Science. This Week in Science. This Week in Science. This Week in Science. Science. Got a laundry list of items I want to address, from stopping global hunger to dredging Loch Ness. I'm trying to promote more rational thought, and I'll try to answer any question you've got. But how can I ever see the changes I seek when I can only set up shop 1 hour a week? This Week in Science is coming your way. You better just listen to what we say, and if you've learned anything from the words that we've said, then please just remember it's all in your head. Cause it's This Week in Science. This Week in Science. This Week in Science. Science. This Week in Science. This Week in Science. This Week in Science. Science. This Week in 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 Science.